Hello, as we continue with the series of microbiology tests. In this session, we are going to look at what we call culture media. And culture media in microbiology lab, these are substances which we use to grow microorganisms, that is bacteria and fungi in the lab, so that we can isolate them, so that we can give right treatment. And as we begin, if it is your first time to be on this channel, please be reminded to subscribe so that you continue to be informed whenever we release the videos. So with the culture medias, we are looking at how we are going to grow microorganisms in the lab using samples from patients so that we can isolate organisms which might be pathogenic for better treatment. And these culture medias contain ingredients such as agar, and agar is a solidifying media or agent. It does not contribute to any nutrient, but its role is to solidify the media. And this agar is got from seaweed extracts, and its role is to solidify the media. Then we have peptone water. This peptone water contains the meat extract, which is acting as a carbon source. It contains carbohydrates, that is polysaccharides, which act as the carbon source, at the same time having the peptones, which provide proteases, the amino acids, as a nitrogen source. So that is the peptone, which is composed there. Then we have also electrolytes and other ingredients. But in this session, we are looking at classification. We want to see how can we classify different culture media in the lab. And we are using only 3 criteria for classifying them, whereby we are having classification according to the consistency of physical nature. Then we have classification according to the function, and a classification according to the composition. So according to physical nature, culture plates are classified to be liquid, to be solid, to be semi-solid. So these are the 3 categories under consistency of the physical nature of the plates. So plates which are liquid in nature, these ones, they are used for growing microorganisms which are in smaller numbers. So if you want to isolate organisms which are in smaller numbers and you want to increase their number, we can use liquid, and sometimes these liquid media, we call them broth. So they are used to culture samples whose organisms are expected to be in low numbers so that they can be amplified or increased in number. And these liquid medias, the oils or the broth, examples here we can give is nutrient broth. We have nutrient broth. We have glucose broth. And then we have peptone water. Another example is peptone water. And nutrient growth contains majorly the peptone and the meat extract. And if you get peptone in the nutrient agar, in the nutrient, then we add the glucose, it becomes glucose broth. Whereas peptone water contains majorly the meat extract and the water. And the meat extract is the one that helps us to get the peptones from it. And these are underlying, or they are the simple medias we can use to culture organisms which are non-fastidious. Then another category of liquid medias, we have also examples of enrichment media like selenite F, is also a liquid media. Selenite F is a liquid media. We have alkaline peptone water and tetrathionate broth. All of those are liquid media. So I want you to remember that apart from nutrient broth and peptone water plus glucose nutrient broth, we also have enrichment medias which are liquid in nature, such as selenite F broth. We have alkaline peptone water together with tetrathionate broth. Also other examples under liquid media. And the most important thing why we use liquid media are to isolate organisms which are suspected to be in a smaller number in the sample. But the disadvantage of liquid medias, it requires sophisticated ways to count the viable number, because the growth of organisms under liquid are identified by presence of turbidity. So we can know that there is growth by looking at the presence of turbidity, which might not be easy to quantify the organisms. And you cannot even isolate organisms according to their growth morphology, since you can only see them by change in the color of the media to turbidity. So it becomes not good compared to the solid media. So the second one is solid media. And if I have liquid media, I can solidify them by adding agar. The agar base, types of agar base, the best one is New Zealand agar, whereas even the Japanese agar can be used as a solidifying agent. And for the solid media, we use agar between 2 to 3%. So if you add 2 or 3% agar in the media, it is able to solidify. This agar we are talking about, it melts at 98 degrees centigrade and solidifies at 42 degrees centigrade. It was first discovered by Robert Koch and improved by his student, that is Petri, and that's why we got a name called the Petri dish. So this is solid medias, and the majority of culture medias are solid in nature. And examples here we can give under solid medias is we have blood agar, we have nutrient agar, we have chocolate agar, we have mannitol salt agar, we have DCA, and many others. So all these ones are solid in nature due to presence of 2 to 3% agar base, which solidifies at 42 degrees and melts at 98 degrees centigrade. Then the last category under here is semi-solid media, and these ones are made by reducing the concentration of the solidifying agent, that is the agar. So here, instead of using 2 to 3%, 0.2 to 0.5% agar is used. And this one gives what we call a semi-solid agar, and their importance, we use them to isolate organisms which are motile. So they are used to detect motility, or the bacteria which are able to move in the media. So you can use a semi-solid media. So that is what we can look under physical consistency. We have liquid for growing organisms in smaller quantities so that we can isolate them. Then solid medias, these are the routine medias we use after adding agar as a solidifying agent, 2 to 3%. Whereas semi-solid, we add 0.2 to 0.5%. Please don't forget that these ones are good for isolating organisms which are motile from those which are non-motile by looking at their ability to migrate from the stub line. That is what we can talk about. Then classification according to composition. Here we are going to see being classified as simple, It can be simple or basal media from the word base. Then we have those which are complex. Then we have those which are defined. You can call them defined or you can call them synthetic, chemically defined or synthetic type of media. So this, according to composition, simple media, as the word suggests, these ones, they are used to grow organisms which do not require special requirements. So they are used for growing non-fastidious microorganisms. These ones which do not require special nutrients. So we can use simple media. So they do not require any extra nutrient to grow. For example, like Staphylococcus can be used here. Examples of like Pseudomonas, which do not require special requirements. So the media contains majorly the nutrient agar, the meat extract, which the nutrient agar has the meat extract or yeast extract together with the peptone. These are the only components which are available, plus the salts such as sodium chloride. And there is no any addition of any extra nutrient. Examples of basal media here we can give is nutrient agar. We have glucose agar. We have peptone is also an example of the simple medias we can use in the lab. Not forgetting also the nutrient broth can also be used. These medias, we are saying they do not have special nutrients and they are used to grow for, so they are basics, they contain basic nutrients for growth of non-fastidious microorganisms. That is what we can note there. Then with the complex, majority of these one of them traits belong here. Apart from the nutrient agar, the nutrient broth, the glucose broth, the peptone water, others are complex. And complex means they are made up of extra nutrients. Here we see they are made up of extra nutrients. So meaning even fastidious organisms or organisms which require special requirements can grow here, or nutrients. And these complex, these nutrients are in unknown quantities. It contains nutrients in unknown quantities. So the quantity of the meat extract is not known, and the or yeast or any other carbon source. So we are seeing the carbon source and the nitrogen source. The quantities are not defined. That's why we are calling it complex, or you can call it undefined media. It contains extra requirements or nutrients with meat extract, with the carbon source, with the nitrogen source, but in unknown quantities. And examples of these complex media, we have the blood agar, we have the maconche, we have the chocolate, we have the mannitol salt agar. All those are complex. We have the XLD, the DCA. All these are examples of traits and are complex, which is different from the defined or synthetic medias. These ones, they contain extra nutrients in known quantities. They are in known quantities. So the one which is carbon source is known. The one for nitrogen source, its quantity is known. For the carbon source, we use glucose or glycerol. Whereas for the nitrogen, we can use ammonium ions. So, but it contains extra nutrients in known quantities. That's why we call it defined. So the difference between complex and defined is that complex, the quantities or the nutrients involved in the meat extract, in the yeast extract are not known, whereas in the defined, they are known. That is how we can get. An example of this defined or synthetic media, example we have is Dubos media. Dubos media is one of the examples of defined media. So this is what we can talk about under composition. So the last way of classifying is by function. And according to function, we are going to see plates such as enriched. We have enriched media. And with enriched media, these media contain extra special requirements. These ones contain special requirements for growth of fastidious. So we use them for growth of fastidious organisms. Organisms that require special nutrients. And these enriched media, they have special requirements such as blood, whole blood, sheep whole blood or human whole blood or horse. It can be serum. It can be egg yolk. So these extra requirements come from this. Examples of enriched media include blood agar. Blood agar is an enriched media. Chocolate is an enriched media. LOJ is enriched media, that is Lawinson-Jenson media for Mycobacterium. Even Lofra's serum syrup. Lofra's serum syrup is also an example of enriched media. So I want you to note examples of enriched media to be blood agar. We have chocolate, which is made by heating blood agar at 80 degrees centigrade, converting it from red to brown color. Then we have other plate agars like the Lofras, the Maconke. All those are enriched media we can use. But with exception of Maconke, we have the blood agar, the chocolate, the Lawinson-Jenson, and the Lofras slope agar is the one we can use. Then that was part A. Part B, about the classification by function. We have enrichment media. Enrichment media. What is the difference between enriched media and enrichment? Enriched media, majority of them are solid in nature, whereas these enrichment media, they are liquid media, or you can call them broth. And these enrichment media, they are enriched with substances that suppress, we can use the word, that suppress growth of commensals. Or you can call them normofloras. So if you get a sample from a site like the GIT, which has much of the normofloras like the Escherichia coli, you can use enrichment media to suppress the growth of these commensals like E. coli, at the same time promoting the growth of the desired organism, like Shigella, like Salmonella, and Vibrio. So it supports the growth of the desired, at the same time suppressing or do not support the multiplication of the commensals so that we are able to isolate the desired organisms. And we are saying they are liquid media. And examples of enrichment media include, we have selenite F, we have an example of tetrathionate broth. Then we have also alkaline peptone water. So these three, the selenite F and tetrathionate, they are being used to isolate enteric bacilli such as Shigella organisms and Salmonella. At the same time, inhibiting the growth of the commensals such as E. coli or the normal flora. Whereas alkaline peptone water is being used to isolate Vibrio cholerae. Vibrio cholerae, we can use alkaline peptone water. So these are the examples of enrichment media. Note the function used to suppress organisms. Those are the normal floras, which are commensals, at the same time supporting the growth of the desired organisms. So they contain substances that do not allow the growth of the normal floras. For example, when using samples from the GIT, from the nose, you require to use this enrichment media so that you only grow organisms which are pathogenic, stopping those which are non-pathogenic. Then part C, under classification by function, we are going to look at selective medias. And under selective media, or culture plates, these are plates. These plates, for them, they inhibit growth of other microorganisms, also at the same time allowing growth of desired organisms. That is what we can say. So they contain substances that inhibit growth of unwanted organisms, at the same time supporting the growth of the desired organisms. So these selective media contain substances like the biosalts, which can inhibit the growth of certain organisms, at the same time favoring the growth of other organisms. Examples include, we have TCBS, we have DCA, that is deoxycholate agar, deoxycholate citrate agar. We have xylose lysine deoxycholate. We have hectone HE agar, that is hectone ambient agar. Then we have LJ. These are some of the examples of selective media, not forgetting mannitol salt agar. TCBS is being used as a selective media for Vibrio cholerae. Whereas DCA, XLD, and HE agars are being used to favor growth of gram-negative bacteria such as Shigella and Salmonella. Then Leucin-Jensen, or what we call LJ media, is used for isolating Mycobacterium tuberculosis. So if you want to isolate MTB, you use Leucin-Jensen agar. Then for mannitol salt agar, it is being used to isolate organisms such as Staphylococcus species. which have ability to grow in areas of high salt content. These ones are known as to be harophytes. So they can grow in 5% salt media. So these are some of the selective media that can be used to enhance growth of desired organisms, at the same time containing inhibitors for the growth of unwanted organisms. And examples, we have seen them here. Then another part we are going to look at is a differential media. And among differential media, you can also call them indicator media, because they contain special indicators such as phenol red, such as neutral red, which are used to isolate organisms according to their growth characteristics. So differential medias are used to isolate or to identify organisms according to their growth characteristics, like change of their color and also the color of the colonies, because it contains indicators such as neutral red or phenol red that are being used to isolate organisms by looking at their growth characteristics such as color. For example, on maconche, if I have maconche, this one contains lactose, so it is used to differentiate organisms which are lactose fermenters from those which are non-lactose fermenter by looking at the change in color, whereby if I look at pink colonies, that one tells me they are lactose fermenters. And if I look at the color, the colonies being colorless, that one means they are non-lactose fermenters. Then another differential is mannitol salt agar. So mannitol salt agar is both selective and differential. Even maconche is both selective and differential. So we can also use it for identifying organisms such as the Staphylococcus species, which are harophyte staph species. And not forgetting that even blood agar is also a differential media, being used to distinguish those which are hemolytic that can be from those which are non-hemolytic, those which are able to hemolyze blood from those which cannot hemolyze. So we see those which completely hemolyze, those which partially hemolyze, and those one which do not hemolyze, such as alpha, beta, and gamma hemolytic. So that one becomes also a differential media. Don't forget that differential media, they are used to isolate organisms according to their growth characteristics. That is such as change in the color of the media. Lastly, in this session, we are going to look at the last part under classification by function, which we are going to call transport medias. We're going to call them transport media. And among transport medias, which is part E, we are calling them transport media. These ones are used to transport delicate organisms. We use them to transport delicate organisms which cannot withstand harsh conditions during transportation. That is, so these ones, we can say that they are non-nutritive. They do not contain any nutrient. Their role is to maintain viability of the organism so that they can reach in the lab in the way they were. So how do they keep them viable? They prevent them from drying because they are liquid in nature. At the same time, they contain buffer that do not allow them to multiply. So they prevent them from multiplication, so that the way they were in the body, they reach in the lab as the exact number. And they contain also some factors that do not allow multiplication of normal floras, so that you can only get the pathogenic bacteria. So the importance of these transport media is to maintain the organisms in the numbers the way they are in a human body. During transportation, we prevent them from multiplying, at the same time maintaining them viable, so that when we culture them, they are able to grow. That is what we call transport media. And examples of transport media, we have Amis media, we have Stewart's media, then also we have Calebrea media. Calebrea is used for transporting samples, that is stool samples, suspected to have Vibrio cholerae. You can use Calebrea. Then Amis and Stewart's, they are used for transportation of samples suspected to have Neisseria gonorrhoeae or Neisseria species. So if you are suspecting Neisseria species like Neisseria gonorrhoeae, you can use Amis media or Stewart's media to transport these organisms without them being undergoing multiplication to increase the number, at the same time remaining viable. So it is important to know the media and the organisms in question whenever we use them, so that they can reach in the lab in a steady state. So this is what we can discuss with you. Another thing I could tell you is that we have been talking about chocolate agar. But chocolate agar, for it, we said it is made by heating blood agar. And it is used for identifying organisms such as Haemophilus influenzae and Neisseria gonorrhoeae. So if you are suspecting those two organisms, the media of choice is chocolate agar, which is got after heating blood agar at 80 degrees centigrade, then it turns to brown. So you can isolate organisms like Haemophilus influenzae and Neisseria gonorrhoeae on chocolate agar. Then for Lofra's serum slope is used for isolating Corynebacteria diphtheriae. Lofra's serum slope, which is an example of enriched media for isolating Corynebacteria diphtheriae. That is what I wanted to add on the enriched medias. So this is what we can discuss with you. We have looked at 3 ways of classifying. That is according to physical nature, according to composition, according to function. And according to function, we have talked about enriched media for organisms which are fastidious and they contain blood, serum, or egg yolk to allow the growth of those fastidious organisms. Examples we have given BA, chocolate, LJ, and Lofra's serum slope. Enrichment media, these are liquid media used to suppress growth of normal floras, supporting growth of the pathogenic. Examples we have seen cyanide F broth, tetrathionate, and alkaline peptone water. We have seen about selective, which inhibit growth of undesired, allowing growth of the desired organisms by containing inhibitors such as TCBS, DCA, and HE, LJ, and mannitol salt agar. Then we have looked at differential, which isolate organisms according to growth characteristics, whereby we have seen McConkie, mannitol salt agar, and blood agar. We have ended with transport media. Thank you so much for learning with us at Medical Sciences. Please, if you have enjoyed the video, remember to like the button. Thumbs up by liking the video, share to many others, and please subscribe and encourage more to subscribe. Thank you so much.